During this Camelot period, a consensus emerged broadly that most clinical trials were too small for reasonable conclusions to be drawn from them. And so in the 1980s and 1990s, more and more trials were undertaken at massive scale. The International Studies of Infarct Survival was one such consortium, spearheaded by the renowned Oxford epidemiologist and Gardner laureate Richard Peto, in some ways the intellectual grandson of Bradford Hill. Their second big study of treatment for acute myocardial infarction is widely recognized as a truly historic endeavor. It used an elegant factorial design, testing chewed aspirin, intravenously infused streptokinase, both or neither. The protocol was deliberately simple, with the result that 417 coronary care units from 16 countries signed up, and 17,187 patients were successfully randomized over the course of 3 years. Each drug worked dramatically to lower mortality, and they worked better together, as the figure clearly shows. However, the question I posed at the outset of this lecture, that question of applicability to individuals, did turn up in the minds of the editors of The Lancet. Trials do give us a sense of what, on average, works for a population of patients. In examining the draft of the trial manuscript, the editors asked whether more subgroup analyses could be undertaken to determine whether some types of patients benefited more than others. The cheeky response of the ISIS-2 investigators has become legendary. They did an analysis by astrological signs to prove the pointlessness and dangers of this type of post hoc data dredging, and in The Lancet published totally implausible variation in the effect sizes for the two interventions based on being a Gemini or a Libra. Now, this was cheeky and very telling. And there's no question that univariate subgroup splicing and slicing and dicing is an invitation to both type 1 and type 2 errors. Underpinning it is what Parker and I have called the assumption of multiplicative effects. And that is, relative benefits are assumed to be constant across subgroups, so absolute benefits are greater for high-risk patients just by simple mathematics. It's a pretty good rule of thumb, but it is also at times quite misleading. Think of it this way. Epidemiologists have rightly warned against inferences based solely on biology when it comes to treatments. That's one of the axioms of evidence-based medicine. But then they make inferences based solely on questionable statistical assumptions, and surprise, sometimes biology bites back. Sometimes it matters a lot. There are actually quite a few examples in the literature, but I'll pick only a telling and recent one. Dalcetrabib is a cholesterol ester transfer protein inhibitor. It dramatically raises HDL cholesterol levels, and it should, in theory, improve cardiovascular outcomes among people who've had an acute coronary event. However, in 2012, a well-conducted trial involving no less than 15,871 patients reported no benefit from the drug. A group of Canadian investigators later found that a single nucleotide polymorphism affecting a gene on chromosome 16 was a massive modifier of the drug's effects, as shown on the slide. The reason the drug appeared useless was because those with genotype AA had a 39% reduction in the endpoint compared to placebo, while those with genotype GG had a 27% increase in events, and heterozygotes fell in the middle. And one has to wonder, how often has this type of response heterogeneity been the reason why a particular drug failed in a randomized trial? And again, logical extension, how much better would treatment be if we could match drug selections more accurately, not just according to genetic markers, but perhaps taking into better account a range of determinants of treatment responsiveness? We want to be clear about something here, and that is that evidence-based decision-making based on randomized trials was and remains a massive improvement over guesswork based on the last case, the best case, the worst case, and other forms of clinical anecdotage. In general, it is perilous to draw inferences from pathophysiology or from observational epidemiology without some convergent validation involving rigorous clinical experiments. But the evergreen challenge of generalizing trial-derived averages to individuals has persisted.